Hello, everyone, and welcome to today's live broadcast, Multiparameter Cell Cycle Analysis, presented by Dr. James Jacobberger, Professor Emeritus and Director of Cytometry and Microscopy Core, Case Western Reserve University. I'm Alexis Krause of LabRoots, and I'll be your moderator for today's event. Today's webinar is part of the Protein and Cell Analysis Education Series, brought to you by LabRoots and sponsored by Thermo Fisher Scientific. For more information on our sponsor, please visit thermofisher.com. Now, let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. So, without further ado, Dr. Jacobberger, you may now begin your presentation.
Thank you, Alexis. Hello, everyone. The term cell cycle analysis has come to define cytometric assays to count cells in cell cycle phases, compartments, or states. I'll use these words throughout the talk interchangeably. This slide shows a single parameter histogram in the upper left for cells stained for DNA content, which gives us three phases, G1, S, and G2 plus M. A two-parameter plot in the lower left shows cells stained for DNA content plus a mitotic marker, which then gives us four phases, G1, S, G2, and M. M is synonymous with the mitotic stages of the cell cycle. G1 are cells with one genome, S is cells that are synthesizing the second genome, and G2 and M are cells with two genomes. On the right, a three-parameter plot shows cells stained for DNA content, a mitotic marker, and two cyclin proteins. These are proteins that oscillate periodically and regulate the cell cycle. These data create a multi-compartment model of contiguous obligate states that cells pass through during the cell cycle, and that is multiparametric cell cycle analysis. In general, DNA is the primary base marker. It provides a three-compartment subdivision ordered in time, G1, then S, then G2 plus M. Periodically expressed genes or activities subdivide these phases further with the goal to create a model with objectively defined compartments that are unambiguous. That is, the cells pass through each compartment unidirectionally once. This slide shows two-parameter histograms of data synthesized from expression profiles of such periodic expression. The upper row contains expression that rises and plateaus within a single phase. The middle row shows expressions that rise across phases. In both rows, cells divide at the plateau level, creating daughter cells that have half that level. In the third and fourth rows, expression rises and declines within a single phase, for example, the first, second, and fourth columns, or across phase boundaries, the third column. These are synthesized patterns based on ideas, but we and others have observed most of these patterns when looking at specific markers. In many cases, the compartments derived from these patterns are not unambiguous. For example, mitotic markers show elevated expression and mark mitotic cells, but the pattern contains cells that are entering and exiting mitosis within the same data space, i.e. the compartment is ambiguous. To render these ambiguous compartments unambiguous, we need additional markers. I now introduce a fixation and staining protocol that is extensively published, so the specifics are unimportant, other than the lengthy time it takes to prepare cells prior to cytometry. Key elements are formaldehyde to fix the cells and prevent further enzymatic activity, followed by alcoholic denaturation and permeabilization, or detergent permeabilization without denaturation. Different markers are optimized by variations in this basic protocol. Since markers are intracellular, optimized staining requires time, which is 30 to 90 minutes depending on the desired data quality, with 90 minutes providing the highest signal-to-noise ratio. Further, after antibody incubation, 3 washes at 15 minutes each plus centrifuge time are optimal. In the next few slides, I'll present data for K562 cells, a human erythroid leukemia cell line, stained for DNA content, phospho-S10 histone H3, which is a mitotic marker, and 2 mitotic cyclins, A2 and B1. We'll walk through the resulting data analysis step by step to create a multi-compartment cell cycle analysis. This slide illustrates the first analysis steps which clean up the data. These include doublet discrimination, exclusion of any anomalies from perturbed flow, and a correction for signal drift. A region is set that includes singlet cells based on the shape of the primary DNA signal pulse. The first plot shows the signal peak on the y-axis and the signal area on the x. Doublets, triplets, and so on have peak heights equal to single cells, but areas equal to multiples of single cells. The middle row shows plots of DNA content versus time. For this run, there are no areas of perturbed flow, but if there were, regions would be used to exclude incorrectly measured data. However, although here the effect is small, there is a continuous decrease in the signal over time. That can be corrected in the same way as compensation is applied. The quality, that is, the CV of the data, does not change over time, so the correction improves the CV of the corrected data compared to uncorrected data. We examine all parameters for this effect and correct if the overall effect is severe enough. This doesn't always happen, and the causes behind it are complex and still under study. Since we are going to move continuously through multi-parameter data space through bivariate windows, I've included this slide to illustrate the overall process. What is plotted isn't important because in the following slides we will go through the process step by step. What is important is the idea that we have moved through, in this case, 5-parameter data space, following the arrows in a manner that does not exclude any combination that would equal a cell cycle state. Or at least that is the idea. In this example, we start at the box labeled begin and end with the box labeled end, which is the beginning and end of the cell cycle. The next step is to separate interphase from mitosis. That is shown in the panel on the left. Phosphohistone H3 and many other heavily phosphorylated epitopes increase dramatically when cells enter mitosis. For this epitope and others, dephosphorylation occurs in late mitosis and continues through immediate early G1. Thus, we can segment all M by gating all 4C cells with elevated phosphohistone H3. In these same plots of phosphohistone H3 versus DNA, we can capture the early G1 cells, shown by the arrow and the word newborn. In these same plots of phosphohistone H3 versus DNA, we can capture the early G1 cells. We can check that measurement by examining cyclin B1 versus histone H3. Because of the decreased background of the small early G1 cells, we can separate early G1 from the rare late M cells that have partially dephosphorylated histone H3. And that's shown in the right panel. The next steps are to isolate G1 proper, that is minus early G1. Both G1 and G2 can be segmented using cyclin A2 versus DNA plots with M cells removed by Boolean logic. The remaining cells are in S. The critical G1S and G2 boundaries can be checked by comparing the G1S and G2 phase fractions of the DNA distribution determined by Gaussian modeling of DNA content with programs such as ModFit to the frequencies of cells determined by color coding logic. Because there is shape to the cyclin A2 versus DNA S phase component, it can be broken into early S, S1, and the remainder of S, S2. The plot on the far right shows the DNA distribution color-coded for G1, S1, S2, and G2. Next, we segment G1 into two states, G1A and G1B. The importance of segmenting G1 into two states, an uncommitted G1A and committed G1B, separates what used to be called the restriction point, which is a commitment checkpoint. The mitotic cyclins are repressed in the uncommitted state by activity of the anaphase promoting complex / cyclosome, or the APC/C, and as it is inactivated, cyclin B1 expression increases. The level is still low and expression is continuous, therefore it is not a great marker, but at present some information can be obtained without adding another marker. The left figure shows how we can use the G1S boundary. S1 is color-coded maroon, defined previously, and the early G1 cells, here colored black, to determine approximately where to place the region boundaries. The panel on the right shows the progression of the states G1A, G1B, S1, S2, and G2 in terms of cyclin B1 levels. We next turn to M phase. We define the earliest M state, P1, as 4C cells with maximum levels of cyclins and rising levels of phosphorhistone H3. That is shown in the upper left on the plot of PHH3 versus cyclin A2. The previously defined G2 cells in green define the critical boundary, and the upper boundary is placed at the cluster edge. Thereafter, gating only on M cells, we plot cyclin B1 versus cyclin A2, and the pattern shown in the upper right is segmented at cluster boundaries. In this plot, P1 and P2 are coincident, and P2 is defined by Boolean logic that excludes P1. When the APC begins to activate, cyclin A2 is degraded, and this is captured as PM because cells in this state correlate with prometaphase. When cells have reached undetectable levels of cyclin A2, the mitotic checkpoint is entered with stable high levels of cyclin B1 and depleted cyclin A2. This state is labeled M because it normally correlates with metaphase. If cells are treated with a mitotic spindle inhibitor, for example, metronidazole, they arrest here and this state will become highly populated. Next, cyclin B1 is degraded and this correlates with the onset of anaphase, but the rate of decay does not correlate well with the sequence anaphase 1, anaphase 2, and telophase. Since these states do not correlate well with morphologically defined stages, we label them LM for late mitosis. LM1 is defined by cells with less than maximum but more than minimum cyclin B1. That is, cells that are degrading cyclin B1. LM2 is defined by cells when cyclin B1 has been degraded to a minimum. Within this state, there are additional states that can be defined by morphology. On a plot of DNA pulse peak versus the DNA integrated signal for phosphohistone H3 positive cells, we observe two clusters in a transition. The reason for this is that if the cytometer is well tuned, the late anaphase and telophase cells look like doublets. This is shown in the lower right panel. The cells with the lowest peak signal, that is, those enriched in telophase, can be further subdivided into a group with maximum phosphohistone H3, that is LM2C, and those with lower levels, LM2D. The figure on the lower left shows that cells divide from LM2C and LM2D, or at least that is the working hypothesis. Thus, in a 3D plot, we can visualize most of the 15 unambiguous states, 14 of which are sequentially traversed by unperturbed proliferating cells. An average cell of this population under the proliferative conditions defined by the environment at the time of fixation moves sequentially from G1NB to LM2C, then optionally move to G1NB or LM2D, then to G1NB. This provides a continuous backbone onto which other markers can be mapped. Now I'd like to introduce the idea of adding information obtained from a different platform. The data are from a laser scanning cytometer. In this case, these are a different attached cell line, but in principle the same samples of fixed and stained cells that were analyzed by the flow cytometer previously could be analyzed in this manner. The plot at the upper left are cells stained for phosphohistone H3 and DNA, and I have set a gate for mitotic cells. The plot on the lower left shows nuclear size on the y-axis, and on the x-axis, the density of cyclin B1 in each cell. Region R10 equals the largest nucleus with the minimum cyclin B1 density. Region R15 equals the smallest 4C nucleus with maximum cyclin B1 density. If we look at the R10 images, we observe that cyclin B1 is cytoplasmic and on centrosomes, but not in the nucleus. Many of the centrosomes are well separated. At the beginning of mitosis, cyclin B1 first accumulates on the centrosome, which then begins to separate, and each will end up at opposite each other and create the poles of the mitotic spindle. If we look at the images in R13 to the right of R10, we can see that cyclin B1 has entered the nucleus for approximately 50% of the cells. And in those cells in which cyclin B1 is still in the cytoplasm, there are 2 centrosomes and most are well separated. If we go to R15, the cells are almost all in metaphase. Cyclin B1 is now cytoplasmic again because the nuclear membrane is broken down, although the cyclin B1 density is still high because the cells have rounded up and cyclin B1 decorates the mitotic spindle. Now I'd like to take a step back and go over the underlying principle behind the complex bivariate data patterns we've been analyzing. This slide shows expression of 2 parameters over time. The top row is parameter 1 and the middle row is parameter 2. The data are events, cells, that vary on the y-axis. That is, for every point in time, there are multiple cells that represent the expression level if all of the cells in the population were synchronous. The 2 parameters are correlated in time. When a proliferating population of cells is sampled, because they are asynchronous, all points along the timeline of expression are represented. Thus, if we plot parameter 1 versus parameter 2, we get 2 parameter histograms as shown in the bottom row that look like typical flow cytometry data. I've color-coded the clusters and transitions, bottom row, and their corresponding segments in the 2 top rows. For example, the 2 stable minima at the beginning and ends of the time sequence are ochre and brown. They are represented in the bottom bivariate plots in the first cluster in the lower left. The sharply rising expression of parameter 1, cyan, is correlated with expression of parameter 2 at a stable minimum, also cyan. Therefore, the transitional cyan cells move, in quotes, to the right in the parameter 1 direction and remain fixed in the parameter 2 direction in the bottom of the histogram. If the expression of one parameter is shifted in time relative to the other parameter, the number of cells populating clusters and transitions is affected, middle and right columns. Now, going back to our data from the laser scanning cytometer, If we calculate the median fluorescence and plot it on the y-axis and plot relative cell cycle time calculated from the compartment frequencies on the x-axis, we can see the profiles underlying the parameters. Here, phosphohistone H3 expression and cyclin B1 density, or any other marker that we have included. And if we have imaging data, then we can do the same thing for other information by showing the time-related cumulative change in frequencies. For example, this plot shows the parameter expression profiles and reveals the variation in entry or passage through the mitotic stages. It can be seen that accumulation of two centrosomes parallels prophase, and both correlate with the rise in phosphohistone H3. Equally, cyclin B1 enters the nucleus rapidly in all of the cells over a very short period, and this is followed immediately by entry to pro-metaphase. The expression of cyclin B1 through this period is maximal and stable, but the density sharply rises at the end of prophase. Metaphase is more variable, but by the time most of the cells have entered, cyclin B1 density remains high. By the time anaphase begins, cyclin B1 density is falling. Thus, we can quantitatively correlate expression, movement of molecules, and morphological changes. This is about where we and others have taken multiparametric cell cycle analysis. So I'd like to make a few comments, and then I'll present some of our recent work. Since I can imagine critics saying that this is all nice, but what real value is this complicated analysis, I'll start on the right side of this slide first. The first practical application is in pharmacodynamics. Cell cycle regulators and other targets that affect the cell cycle are still rational approaches in cancer chemotherapy. This type of an assay might provide improved information in pharmacodynamic studies and subsequently as a possible therapeutic guide. We have built an analysis of DNMT1, a target of azacitidine or decitabine therapies, using a reduced version of this approach. Second, for any blood or bone marrow analysis, some subset of this approach could reduce the complexity of data by normalizing expression of other molecules across the cell cycle, possibly reducing the width of data by a factor of 2, and coincidentally providing high-quality doublet discrimination. Finally, my good friend David Headley believes that it is time to use this information to study the effects of drugs in therapeutic studies using xenografts. I believe he's right. To the left side, these are the things that I would do going forward. G1 can be compartmentalized just as we have done for mitosis. There are many candidate markers. The data will be a little more messy because G1 is more highly variable, but I expect it to break apart in a similar fashion. G2 has a major DNA damage checkpoint operating. This has been extensively studied, and the probes are available to see if G2 could be as interesting. I would like to do studies that incorporate data from multiple platforms into a single correlated data set using the principles I've outlined. Imaging is my favorite, but slit scan is underexplored, and the work of George Brattain and Sergei Gulnik demonstrate that differential permeabilization may be a fruitful way to get at molecular sequestration. I think the day has come for a new generation of probes, and camel antibodies are my favorite in that regard. Multiparametric data should be analyzed by probability state modeling. See Bruce Bagwell's work on this. This would eliminate or reduce significantly the number of decisions about where to set regions. Finally, I think improved hardware is not out of the question. Speaking of improved hardware, I'd like to present some results that I'm very pleased with. Here's a reference to a paper by Goddard et al. On acoustic focusing cytometry. This is a technology where acoustic energy is directed into the flow cell in such a way that the center of the stream is a volume where the waves cancel and there is an energy absence. Thus, the cells move to the center, and once in the center, they stay there. This is illustrated in the next few images by an illustration on the left and a photograph on the right. Thermo Fisher has commercialized this technology in the Attune Next flow cytometer. What this meant to us is that we could try to develop a semi-washless protocol and reduce the time that we use to stain cells. The idea is to wash away the fixative normally and then incubate in antibodies in a small volume, then add one large dilution. The Attune instrument can run a large volume through rapidly, up to 1 ml per minute. The instrument can do this because the cell will all move to the center and remain in focus. Thus, the unwashed large volume sample might be able to go directly on the instrument after an incubation period and a dilution. Here's the protocol. Without going through the entire thing, it is just like our standard protocol, except we save time on the washes and acquisition time with a time savings of 2 hours, 5.5 hours compared to 3.5 hours. Additionally, in these assays, each centrifugation results in the loss of cells. To the extent that starting with 1 million cells usually provides significantly less than 100,000 analyzable events. We have not quantified it, but this washless assay does provide a noticeably better cell recovery. We'll now look at some comparative data. Here are the data comparing the two assays. They are visually equivalent. The top row compares phosphohistone H3 versus cyclin A2. The middle row compares phosphohistone H3 versus cyclin B1, and the bottom row compares cyclin B1 versus cyclin A2 for mitotic cells. We quantified 5 mitotic states as these are the rarest events. The regions are shown in the bottom row. Here are the results. There is not a significant difference between the percentages of cells within each compartment or the levels of cyclin A2 and B1 in the cells. Therefore, this is a significant step forward for this kind of work. And since I have made the comments that I would ordinarily save for the summary, I'll stop here. One more thing. The work shown in this talk depended on the work of many people over many years from my own laboratory and those of many others. The references listed on this slide are reviews of the work with extensive references to our work and that of others. Thank you for listening. The people who did the work at Case Western Reserve University are Tammy Steffen and Phil Woost on the experimental side, Mike Stromkowski and Allison Kipling on the instrument side. From Thermo Fisher, Suzanne Schloman ran our initial experiments, and Brian Wortham provided access to the instrument. Jolene Bradford and Mike Ward were critical for moving this project forward, and we continue to work with them on other projects.
Thank you, Dr. Jacob Berger, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type them into the Ask a Question box and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. Questions we do not have time for today will be answered by Dr. Jacob Berger via email following the presentation. Our first question is, presumably, information increases with the addition of parameters. Is the relationship simple? And if so, what is the relationship between information and parameter number?
I've looked at this in some detail, and the short answer is that there's no simple relationship. I have the feeling that we hit pay dirt early on with parameters that were more informative per parameter than I expect to get per parameter in the future. But for sure, a parameter, an additional parameter should add at least one piece of information, or it's redundant. And it's probably not unthinkable that 2 or 3 pieces of information might be added per parameter. That's about all I know about that.
Our next question is, you have presented an analytical scheme as a backbone with the idea that this is indefinitely extensible. Have you tested this idea?
The short answer is no, I haven't. There's no reason that I can possibly think of that would prevent that from being true. So theoretically, it's doable, but a real test of it is still needs to be done.
And it looks like we have time for one more question. Can you give some examples of when a no-wash or minimal wash protocol will be effective?
Yes, I think well, as far as I'm concerned, it's effective almost always because it saves time. But I think the perhaps more important case would be for things like clinical samples that are very limited in cell numbers. So the idea of then not losing cells through centrifugation comes to the forefront, and the idea of incubating with antibodies and then just diluting up and running and being able to run almost the entire sample is very appealing, especially for clinical samples. We did a study here recently that was published recently on sickle cell disease and treatment of patients with decitabine, low-dose decitabine, to reactivate fetal hemoglobin. And what we were looking for was the decrease in DNMT1 as a function of the decitabine dose. And all of the samples had been done prior to us developing the assay, and we didn't have any input into how the samples were prepared. Those samples I don't remember the exact cell numbers, but it was very few cells, something like a couple of 100,000 in a relatively large volume of fixative. So precious samples that where we really didn't want to lose we didn't want to lose any cells. And so in that case, the no-wash approach is almost a lifesaver. It's not that it can't be done with washing, but I'm certainly much happier with the no-wash solution.
Thank you again, Dr. Jacob Berger. Do you have any final comments for our audience?
No, I hope you enjoyed the seminar, the webinar. My email is, I think, in the credits or whatever, and I'm happy to answer any questions if somebody has them after the fact through email.
I would like to once again thank Dr. Jacob Berger for his presentation. I would also like to thank Labbers and Thermo Fisher Scientific for making today's educational webcast possible. Questions we did not have time for today will be answered by Dr. Jacob Berger via email following this presentation. Before we go, I'd like to remind everyone that today's webcast will be available for on-demand viewing through May 30th of 2018. You will receive an email from Labbers alerting you when this webcast is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.